Now we are going to see about outbreeding devices.
Okay.
So what is generally outbreeding devices? So we have seen about pollination, right? In pollination we have seen self-pollination. Yeah. So autogamy is nothing but self-pollination, where the pollen grains of the same flower come and reach the stigma of the same flower. Okay. So that autogamy, if you see that one is a disadvantage or advantage, means that autogamy is disadvantage to plant. Do you know why? Because it creates various inbreeding depression in plants. Inbreeding depression means because the same genetic character. Okay. So continued self-pollination, it leads to inbreeding depression, like production of less flowers. Those plants will produce less flowers, and they'll be having some disadvantages. Okay. So in stigma receptivity and everything, they'll be having some disadvantages in production of non-viable sterile plants and everything. So that inbreeding depression is nothing but continued self-pollination. Self-pollination, if when occurred once or twice, it is okay in the same plant, but it is occurring continuously. Continuously the plant is self-pollinated. It causes inbreeding depression in those plants which is pollinated by these plants. Okay. So inbreeding depression is nothing but the production of less functioning plants, flowers. Okay. So that to come across that depression, these plants itself, they grow in order to overcome this self-pollination. Like stamens will be away from the stigma, and there will be some plants produce only unisexual flowers, only male flowers, and only female flowers will be produced in different plants. So some plants come across this, their motive is to overcome self-pollination. Okay. So they should not, their self-pollination should not take place. So what all the plants should do, they are making, like they are producing a large distance between the pollen grains and the stigma, and only unisexual plants flowers are produced and everything. Okay. So they are making that the pollen of the same flower should not come into the pollen of stigma of the same flower. Okay. So that is the motive of the plants, to overcome inbreeding depression. So that is the outbreeding devices. Outbreeding devices means the devices produced by the flowers to overcome inbreeding depression. That is known as outbreeding devices. Okay. So for example, also by inhibiting the pollen tube inside the pistil. Okay, the growth of pollen tube is inhibited by the pollen of the same flower. That is also a kind of inbreeding device. Okay, that is also outbreeding device. So so now we will be seeing about pollen pistil interaction. Okay. So this pollen pistil interaction, how can you see that pollen pistil interaction is nothing but, so pollen is nothing but the pollen grains. Pistil is nothing but the female gametophyte, which consists of stigma, style and ovary. So this stigma of one flower can be filled with pollen grains of various flowers. What all the pollen grain coming on the air, what are the dust particles and everything will be placed in this stigma. So even so this stigma has to correctly identify the pollen grain of the same species. Can you understand? For example, the flower of mango, flower of banana, flower of guava. Okay, two flowers are there. So the flower of mango has stigma. Flower of guava has stamen, has pollen grains. Can the pollen grains of guava plant come and reach the stigma of mango plant? No. The pollen grains of only mango plant can reach the stigma of, it reaches the stigma of mango flower. Okay, but also this stigma of guava flower also reaches the stigma, reaches the pollen grains of guava flower. It also reaches the stigma of mango flower. Okay, but this mango flower has to select the pollen, has to select the pollen grains of mango flower only. So therefore therefore some chemical substances which reacts there to accept only the species where this flower belongs to. Only mango plants accept the pollen grains of mango flower. Guava plants accept only the pollen grains of guava flower. But various pollen grains are present in the stigma. Guava, mango, apple, so various pollen grains are present in the stigma. It should select the species which it belongs to. So that is known as pollen pistil interaction.
If it is selected wrongly means
No, it cannot select wrongly. It should select only that
only that one. Okay.
So for that only many chemical reactions are taking place there. Okay, to recognize the pollen. Okay, pistil has the ability to recognize its pollen. Okay, so and after recognizing, it promotes post-pollination changes, like after pollen grain enters the stigma, after the selection of the pollen grain is present there, those pollen grains will develop pollen tube. In that pollen tube, two male gametes will be developed from a generative cell, which we have already seen. So that three-celled stage which is released, it is shed by the pollen. So this is the pollen. So here generative cell, here vegetative cell, which we have already seen. We have already seen in the previous session. So this cell will be covered with the cytoplasm, This cell again divides into two, so therefore there will be 2 and 1. So these two known as male gametes. So these male gametes come in the pollen tube like structure, and they will be in the pollen tube, these two male gametes. Okay. And now they go in. So by reaching the stigma of the carpel, the cell will be in this structure. After entering to the stigma and entering to the style, so this is the style. While entering to the style, they will develop a pollen tube like structure. This is known as tube with 2 male gametes. So these will be going here, and these here the ovary will be there, and they'll fuse with the ovary. Okay. So this is the pollen pistil interaction. Okay, it is taking place. So there are various chemical components which are reacted to that various pollen grains which are present in the stigma by the same flower to identify the pollen grains of the same species. Okay. So mechanism which is growing there. Okay. So now so there are 2 male gametes here. Okay. One male gamete fuses with the egg cell. Okay, egg cell is present already. We have seen the structure in the previous section. So here the 2 synergids will be there. Here egg cell will be there. Dipolar nuclei. Okay, will be single cell with 2 nuclei. Okay, and here 3 antipodal cells will be there. Okay, so these synergids will have some filiform apparatus in them. Okay, so this filiform apparatus leads the male gamete to reach the egg cell. Okay, so one male gamete fuses with the egg cell and forms zygote, and the other male gamete fuses with the dipolar nuclei, and it becomes 3 in number, and it has 3 in number of chromosomes, which is triploid, and it becomes the endosperm. Okay, so this is the pollen pistil interaction. Okay. So it is a dynamic process. Okay, it is a dynamic process, is nothing but it recognizes its own pollen from the same variety, and promotion or inhibition of the pollen. It has to promote or it has to inhibit. Okay, so that is the dynamic process which is occurring there. So next we are going to see about artificial hybridization. So what are the methods which are done in artificial hybridization generally? What are the method which is done in artificial hybridization? So there are 2 methods are done: emasculation, bagging. So artificial hybridization is nothing but by the use of human beings. Okay, the human beings, they regulate the pollination process. Okay. How they regulate the pollination process? So emasculation is nothing but in a flower. Okay, so in a flower. Okay. Okay, wait, I'll say you. So emasculation is nothing but in a flower, both anthers and the carpel. Carpel is nothing but stigma, style and ovary. So these both are present. Anthers also present many, and the carpel is also present. So emasculation is nothing but the cutting out of all the anthers with the forceps. Forceps is nothing but scissors. Cutting out of all the anthers of the flower, except the carpel, female gamete. Okay, why they are cutting all the anthers? Because to prevent these pollen grains to fall in the stigma of the flower. Can you understand? It prevents the pollen grains of the same flower to reach the stigma of the same flower. Okay, so this they will be doing this in the bud stage itself. So therefore the stigma will be clean now without any pollen grains. Okay, so therefore the anther is cut down. So therefore no pollen grains to reach the stigma of the same flower. Okay, so that is known as emasculation. Emasculation is nothing but the cutting down of anthers in the bud stage of the flower. In the bud stage itself they will cut the anthers. Okay, so therefore the stigma will be free of pollen grains. So now bagging, we have to ensure that no other anthers from outside the environment should reach the no other pollen grain in the outside environment should reach the stigma of the flower. They have to ensure, no, so they bag the carpel, they bag the flower. Okay, so therefore by bagging they can ensure that no pollen grains from the outer environment reaches the stigma of the flower. So therefore the stigma of the flower is clean now. So what they do, they introduce their needed pollen grain of the same species, whatever the pollen grain is.
The species and a new plant.
Yeah, new plant is produced. Okay, so they'll be taking pollen grains from a well developed plant which is producing more fruits and everything. So they'll collect pollen grain from that plant and they'll be giving that pollen grain into this bagged flower. So therefore they'll produce seeds and everything. So that is known as artificial hybridization. We are cutting down the anthers, we ensure that no pollen grains engage with the stigma, and we bag the carpel, bag the flower, okay, with the sheet or something, and the sheet or a bag. Okay, so now after bagging they get their potential pollen grains and they put it in the stigma of this flower. So that is known as artificial hybridization. Okay. So double fertilization we have already seen now.
Yeah.
So double fertilization is nothing but the fertilization of that there are two male gametes. Okay, in the pollen grain there are two male gametes when they enter the style.
Yeah.
One male gamete fuses with the egg cell. Okay, and one male gamete fuses with the egg cell and becomes diploid chromosomes, and it has diploid chromosomes. It becomes diploid. And the other male gamete, second male gamete, fuses with the center dipolar nuclei, and now it is triploid. So this triploid cell becomes endosperm, and now this diploid cell, it becomes embryo, it becomes zygote. So since the two fertilization of first male gamete, fertilization of second male gamete taking place here, it is known as double fertilization. So that is only known as double fertilization, okay, generally. Okay, and now the triple fusion, the triploid cell, it is known as primary endosperm nucleus. Okay. So after zygote the embryo is formed. Okay, so this kind of fertilization is also known as syngamy, where male gamete and female gamete fuses. So that is known as double fertilization. Okay, so now we are going to see about post fertilization structures and events. So after fertilization, what is happening? Okay, we are going to see. So after double fertilization, one male gamete fuses with the egg cell and becomes zygote, and the other male gamete fuses with the dipolar nucleus, becomes endosperm. Okay, a primary endosperm nucleus, dipolar nucleus. So this is the post fertilization. We are going to see about, we'll first see about endosperm. Okay, so generally this endosperm develops before the development of zygote, zygote or embryo. Why? Because this endosperm nourishes the zygote by giving nutrients and everything. Okay, this triploid primary endosperm nucleus, that triploid, 3 in number of chromosomes, they nourish so they nourish the zygote. So therefore the endosperm is developed, it is formed before the development of zygote, okay, to nourish the zygote. So this endosperm nucleus can be of free nuclear division also. They can continuously divide in the same cell itself, okay, where we saw that development of
we saw coconut, cucumber and
no, cucumber is in the other example.
yeah, that's other example I'm saying.
So free nuclear endosperm is done in coconut, milk tender of coconut. So the milk of coconut, you know, so that one is a free nuclear endosperm. Okay, so made up of thousands of nuclei.
Okay.
Okay, so this milk coconut is surrounded by white kernel in coconut, which you use as coconut. So inside some milk will be there, right? Some watery substance milk. So that is free nuclear endosperm. Endosperm that gives nourishment to this white kernel.
Okay.
So that is the endosperm. It undergoes a number of nuclear divisions without the cytoplasm formation. Only the nucleus divides continuously. So that is known as free nuclear division. Okay. So that is known as free nuclear endosperm. So this endosperm can be completely consumed by the developing zygote, or it can be remained the same. So completely consumed in plants like example pea, groundnut and beans. So pea, groundnut and beans, they will completely consume the endosperm. They will be completely consuming the primary endosperm nucleus for their development. Okay, and it may persist until the seed formation. The endosperm can persist in some plants like castor and coconut. So this endosperm persists in coconut, castor and everything. So they do not completely consume that. Okay. So this is about endosperm. Okay. So next we'll see about embryo.
Okay.
So this embryo, we have seen dicotyledon embryo, monocot embryo. In monocot plants, dicot embryo and dicot plants. So dicot is nothing but they have two cotyledons. They have a plumule which gives rise to shoot, and radicle which gives rise to root. Up below the radicle they will be having a root cap, and hypocotyle, two cotyledons will be there, epicotyle and everything. And in monocot plants they will be having only one cotyledon, that is known as scutellum. Okay, scutellum mainly in grass it is known as scutellum. And they have embryonal axis, and they have a covering to the plumule called coleoptile, and the covering of the root radicle is known as coleorhiza. So that and all we have already seen in the previous chapters and everything, and they have shoot apex and everything. Okay, so this is about embryo. Okay, they have hypocotyle. Hypocotyle is nothing but the middle part in the dicot plants. So they undergo various maturations, okay, like a globular embryo, heart-shaped embryo, and they become a matured embryo. So they undergo various changes, okay. So while changing these antipodals, they will degenerate. Antipodals will degenerate, and the synergids, the two synergids, that also degenerate. So only the egg cell and embryo will be remaining. So they undergo these developmental changes, okay. Antipodals which are at the top, they will also be degenerating, okay. So next we'll see about seed. Okay, so next session we'll be seeing about seed. We are going to see about seed.
Okay.
So seed which they persist with the embryo which is endosperm nucleus, they are known as which do not persist, that is totally consuming the endosperm. It is known as non-albuminous seeds. Okay, so no albumin in them, so it is known as non-albuminous. For example,
Albumin means
the energy, the nutrients, okay, which is given by the endosperm. Okay, so the non-albuminous, it is known as non-albuminous. Example, pea and groundnut. Okay, and those seeds which retain their
nutrients
endosperm
and the energy.
it is known as albuminous. Okay, and the examples are wheat, maize, barley, and castor. Okay. And the next one, some seeds even persist nucellus in them. Nucellus are nothing but the surrounding tissues which are surrounding the embryo. Okay, tissues which are surrounding the embryo, which is like apparatus and everything. So in some seeds, those nucellus is also persistent. Okay, those nucellus is also present in them. Now those seeds are known as perisperm. For example, black pepper and beet. So they have nucellus in them. Okay, nucellus still in the seed form that we discussed. Okay. So and now, so the integuments only generally form the hard coating of the seed. Integuments which are present outside the cell, outside the egg apparatus, it produces a hard coating which is known as seed coat. Okay, and the micropyle and everything remains in the seed coat itself, which facilitates the entry of oxygen, the micropyle. And everything, okay, and water content also. So the embryo, that is the seed, may enter a state called dormancy. Okay, dormancy is nothing but for which the seeds are waiting for favorable conditions to germinate. Okay, can you understand? The time period.
We have already saw dormancy, I think so.
No, we have not seen.
Not of the seeds. Okay, continue.
And we will be discussing it later. Okay. So, we have seen about pollen grains. So the viability of pollen grains, liquid nitrogen, NPK, that is about pollen grains. Here in seeds, so the seeds have dormancy period, okay, certain dormancy period where it is waiting for favorable conditions to germinate. Okay, so that period is known as dormancy period in seeds. Okay, and now if you see fruits having various number of various number of tissues in them, fruits generally. Pericarp. So pericarp consists of ectocarp, endocarp and mesocarp. So these are many times fleshy in case of mango, and it is mango, orange, guava. So it is fleshy. So whereas in groundnut it is dry, groundnut, mustard and all it is dry. So according to the layers, according to the characteristic of that fruit, it is divided into the various classes like cone, pepper, fleshy, berry and everything. It is divided. These fruits are divided. Okay. And there are some fruits known as true fruits and false fruits.
What is true fruits and
Yes, true fruit, the false fruits are nothing but those fruits which retain sepals in the fruit itself. In the fruit, the sepals will be remained, the sepals of the flower will be remained. Or the petals of the flower will be
Can you
remained.
give any example?
Wait, the petals of the flower will be remain and transform into fruit. So like that there are various examples that other floral parts of the flower remain in the fruit also. So that is known as false fruits. Okay. Whereas in true fruits, those do not remain. So maximum number of fruits are true fruits. Only it is developing only from ovary, and the other are weathered off. So maximum number of fruits are true fruits. And the examples of false fruits are apple, strawberry. So apple, strawberry,
This should be there. No?
in apple the thalamus will be there. The thalamus transformed into the apple. Okay, the thalamus which is under the ovary, the thalamus structure, it transforms into the fruit in apple. Okay, in apple and strawberry. So various fruits are some fruits are false fruits, but various number of fruits are true fruits. It is developed only from ovary. Okay, and some fruits are parthenocarpic fruits. Parthenocarpic fruits are nothing but those fruits which develop from only one gamete, only from female gamete. It does not undergo fertilization. It does not undergo fertilization of the embryo, so therefore no male gamete will fuse with the egg cell. It itself develops into fruit. So that is known as parthenocarpic fruits. Example, banana. Okay, so these banana and some various fruits like grapes. So they are all when they are all applied with growth hormone, say they become seedless. Okay, so this is widely used in it is widely economically used. Okay, application of growth hormone to bring out seedless fruits like grapes and papaya and everything.
That is done by people.
That is done by growth hormone, application of growth hormone. They apply using those growth hormones. Okay. So are you clear with parthenocarpic fruits? So parthenocarpic fruits are fruits which
Only female gamete.
okay, only the female, no male gamete is involved. Only the female produces, with the egg itself it produces the fruits. Which is, for example, it is banana. Okay. So here seeds are pollination, and seeds are generally present inside the fruit in angiosperm. So we are seeing only angiosperm, flowering plants only we are seeing. So the seed having hard coating, and it enables very successful reproduction rate and everything. Okay. So here there are various time period of dormancy. We have already seen that. The time which favorable conditions is. The dormancy we have already seen that where the seed is waiting for favorable conditions. So this time period varies for different seeds.
Okay.
Some seeds may wait for only for little time. Okay, and they do not have and they will not be capable of germinating. But some seeds they will be having a long time period where they can germinate even after that time period also. So for example, if you take for hundreds of years also it can germinate. For example, lupin, okay, lupin seed which is produced from Lupinus arcticus in Arctic tundra region, okay. So they have a seed dormancy up to 10,000 years. 10,000 years the seed can be, after 10,000 years of this plant also the seed can grow. Okay, so they have seed dormancy up to 10,000 years. Lupin, okay. So it is found in the region where Arctic tundra. Okay, that region is known as Arctic tundra. Okay,
And seed will not dry in the sun also like that.
It has a long period of dormancy. Okay. So and 2,000 years also a seed is viable, like a date palm. So the seed of date palm, it undergoes a dormancy period of 2,000 years. Okay. So it was found in the King Harold's palace near Dead Sea, this date palm. Okay, for 2,000 years. Okay, after 2,000 years, this date palm it is viable after 2,000 years also. That is the seed which is found in the King Harold's palace. Okay, near the Dead Sea. Okay, and some fruits like orchid, they have various number of seeds in them. They contain thousands of tiny seeds in one fruit. In single fruit, thousands of tiny seeds will be present in the orchid, and so it is also in parasitic species like Orobanche and Striga. So they also contain Orobanche and Striga. So they also contain many number of seeds in them. So there are various number of seeds, thousands of seeds in a fruit. Okay, this is an example. And next we are going to see about apomixis. Okay, apomixis. Apomixis is nothing but the development of seeds without fertilization. It is known as apomixis. Only female gametes evolve. They produce
Like we saw in parthenocarpic.
So parthenocarpic fruits, and what is the difference between parthenocarpic fruits and apomixis? So there the production of fruits without fertilization. Here production of seeds without fertilization. Okay, can you understand? This is the difference between parthenocarpic fruits and apomixis. Apomixis is the production of seeds without fertilization. Okay. So, for example, Laceraceae family and grasses. So they produce seeds without fertilization. Okay. And if you see in the cells of citrus and mango, okay, in the fruits of citrus and mango, the seeds of citrus and mango, the nucellar cells which is surrounding that nucellar region which is surrounding the embryo, they also develop into embryo sometimes. Okay, they also develop into embryo. They also produce seed. The nucellar cells, they differentiate to produce seeds in mango and citrus. Citrus is nothing but orange varieties or lemon orange varieties. Okay. And in some seeds there are more than one embryo is present, which is known as polyembryony. Okay.
Like example is orange.
Orange. It is polyembryo. Okay.
Orange or citrus? Orange.
Yeah, citrus.
Citrus family here also.
Not altogether citrus, only orange. Okay. That is the polyembryony. Example of polyembryony is orange. Okay. If the seeds from hybrid plant is nothing but the hybridization, artificial hybridization, where the seeds are produced from that, they retain the characteristics of the plant. They retain, if the plant produces large number of fruits, the seeds also produce large number of fruits. Okay, so they retain the characteristics of their parent plant. Okay. So this apomixis process, where seeds are developed without fertilization, these are widely used in various regions and it is researched in various laboratories because this is so efficient. Okay, because the seeds are produced without fertilization, no other need to come and fertilize, no other pollination.
No dependence. There is no dependence.
So there are various researchers are going on apomixis all over the world. Okay. So this is about this chapter, sexual reproduction and flowering plants, where we saw about asexual reproduction and sexual reproduction. Okay.
Thank you for watching.
Thank you for watching.
If you like this video, share this video.
If you like this video, please subscribe. And if you have any doubts, please leave them in the comment section. And don't forget to click the bell.
Don't forget to subscribe. Click the bell and sign up.